And I think we're ready when you are.
Okay. Well, good morning, everyone. Thanks for showing up virtually to be here for Reagan Mason's Thesis defense, which is entitled The Impact of Temperature on the Spread of Stony Coral Tissue Loss Disease. Really excited that Reagan's doing this today. She grew up in Old Saybrook, Connecticut, and attended Christopher Newport University in Newport News, Virginia. And she did her undergrad there on a BS in organismal biology. And while she was there, she also did a research internship, working with coral disease at Mote Learning Lab in Florida. And I think that's how she kind of got into this whole field in general. And she's now working at the Smithsonian Marine Station, working under a postdoc looking at the impact of hypoxic multi-stressor environments on corals. That's pretty cool as well. So she's already found a path even before she's fully done, which is great. So without further ado, Reagan, if you want to take it away, I think you can just probably share your presentation mode and then everyone can see it.
All righty.
Oh, wait, sorry, I should say. So how this is going to go, Reagan will do her presentation. Then once she's concluded, I'll applaud her greatly. And then we'll have time for questions from the general audience. You can just raise your hand in Zoom and we'll try and call on you. The committee can ask questions at that time, but the committee will have time after that privately to talk with Reagan, and address any specific questions. So we can also just hold our questions as well. So that's pretty much it. So, sorry, Reagan, go ahead.
All right, sounds good. All right, like Tyler said, my name is Reagan Mason, and welcome on over to my thesis defense, where I looked at the impact of temperature on the spread of stony coral tissue loss disease. So before we get into the nitty-gritty of it, a brief overview of what's going to happen. I'll have an introduction so that we're all on the same page, knowing where this experiment came from, the methods, so what I did, how I did it, results in discussion, so what I found and what those numbers mean, and then a conclusion to tie it all up with a nice little bow at the end. So first up is that introduction. We're going to talk a little bit about coral reefs, Orbicella faveolata, warming waters, stony coral tissue loss disease, and then disease and thermal stress combined. So first up is those coral reefs. They're really important environments, not only to marine life, but also to people as well. So they're lifelines for over 500 million people across the world, particularly along tropical coastal regions. And they're also really important to the oceans. Over 25% of the ocean biodiversity relies on the coral reef at some point during their life cycle, which is especially impressive given that coral reefs cover less than 0.1% of the ocean floor. So now that we know why they're really important, we need to know a little bit about kind of what they're building from. So Orbicella faveolata is one of those corals that builds the structure of the reef. And we're going to talk a little bit about what it is and why it matters. So that top picture there is a colony of Orbicella faveolata out in the wild over in Trunk Bay on St. John. And that bottom piece there is a fragment from my experiment that was collected from Brewers Bay on St. Thomas. So as you can see, there's a little bit of range in colors from that greenish brown in the top picture to the darker brown in the bottom. And so that color actually doesn't come from the coral itself. It comes from a symbiotic algae that lives in the tissue of the coral and provides the coral some nutrients and gets a safe home in turn. And so the other neat thing about corals, and specifically this coral, also as many scleractinian or those reef-building corals, is that it's not just one animal. Each of those little polyps, the little star-shaped things on the corals there, the little dots on the top picture, Each of those are an individual, and then they come together and create a colony that are actually clones of one another. And then diving a little bit more into Orbicella faveolata specifically, which I will probably call OFAV during this presentation. So it's one of 3 sister species, along with Orbicella annularis, which I actually used as my donor coral, and then Orbicella franksi. They're also ecosystem engineers. So like I mentioned, they build that structure of the coral reef. And so they're really important for all the critters that like to hide in the nooks and crannies. They're what make that happen. Orbicella faveolata is also one of the most common Caribbean coral species. So in 2008, the International Union for the Conservation of Nature listed them as endangered on their red list. And then in 2014, the Endangered Species Act for the good old US of A listed them as threatened. So moving on a little bit to warming waters and corals, what could possibly go wrong? So that top picture there is a bleaching coral. So what happens when the corals get too warm or too stressed out is they actually expel that symbiotic algae from their tissue. And because it's a symbiotic algae that gives them the color, you can see on the left-hand side of that top photo, it's very, very white. So that's where it's let go of that algae. And then that bottom picture there shows the temperature difference from the average from 1951 to 1980 as compared to 2022. So as you can see, the temperature difference, it's very much on the warm side. And even though it doesn't look like large numbers difference, it actually makes a really big impact, especially in these ocean communities. So a little bit more into thermal stress, mass bleaching events, so large scale versions of what we saw with that top coral in the last slide, were unheard of before the 1980s. But since then, they've kind of started becoming not quite normal, but somewhat regular. In the 1980s and throughout the 20th century, they were really closely linked with the El Nino Southern Oscillation, a periodic temperature fluctuation. But that's kind of changed since then. During the 2005 bleaching event in the Caribbean, over half of that warming could be directly attributed to climate change. So it's gotten away from that periodic fluctuation and become a lot more regular. The number of years between the recurrent severe bleaching events has actually decreased 5-fold in the past 4 decades. So not only are the corals dealing with these stressors more often, but they're also given less time to recover from these stressor events. And so that's causing a lot of problems for the corals as we continue on. Moving on over to the second stressor in this is stony coral tissue loss disease. And we're going to take a closer look at that. So on the right-hand side there, you can see 2 of my corals from this experiment, both of which are infected with stony coral tissue loss disease, or SCTLD. So a little bit about the characteristics of the disease. The lesions can be either focal or multifocal. So as it spreads, it can infect the coral in one place and just spread out from there. Or there can be multiple lesions on a single colony that kind of spread out and come together toward the end there. There is also denuded skeleton, so just the raw skeleton appearing alongside healthy tissue, or what looks like healthy tissue. So normally as corals die, algae will kind of grow over the skeleton and you won't see quite that difference and that skeleton right next to the tissue. But because of how rapidly this disease progresses, you actually see the tissue right next to the coral that has died. And then also, this disease progresses super, super quickly. For example, for that chunk of tissue and the top side of that coral to get from totally looking fine to that point took only 36 hours, which for corals that can take over 10 years before they start reproducing, it's a really, really rapid time frame. A little bit more about the disease. It's actually associated with a disruption between the host coral and the zooxanthellae, the algae that's living in it. Transmission has been shown to be through direct contact, sediments, commercial shipping, and just suspension in the water column. So we're finding it spreads in a whole multitude of ways. And then finally, the pathogen for the disease is unknown. Coral diseases in general tend to have kind of a team effort as they go. And for this disease in particular, we know that there's a bacterial component. A couple of families have been identified to be present in all of the cases of SCTLD looked at. And we know that antibiotics can be effective at preventing the disease or preventing the spread. And there's potentially a viral component as well, but we really don't know what is causing it at the end of the day. So looking a little bit more at the spread of the disease through the Caribbean, in particular the Northern Caribbean for this timeline. SCTLD was first identified in the Miami-Dade County of Florida over in 2014 off of Virginia Cay. In 2016, the disease had spread to the upper Florida Keys down from the Miami County area. By 2017, it had spread through the middle Florida Keys. And by 2018, it had spread to both the lower Florida Keys and the Mexican Caribbean. And by the end of 2018, it had actually made its way all the way to Cozumel in Mexico. In 2019, the disease spread to Key West, St. Thomas, and Grand Bahama. By 2020, the disease had spread over to St. John, the British Virgin Islands, and St. Croix as well. By 2021, it had spread to the Dry Tortugas, which is a lot closer to Key West than some of these other places, but they have some funky currents that happen over there, and that's why they think it took so long. And then by 2022, it had spread to New Providence Island in the Bahamas. And then as of now, it's spread as far southwest as Nicaragua and as far southeast as St. Vincent and the Grenadines. So it's made its way really through a lot of the Caribbean rather quickly for a coral disease. So looking specifically at the northern US Virgin Islands, it was first spotted off Flat Cay in St. Thomas, over on that southwestern edge there, right by the university. And it stayed mostly contained to that western side of St. Thomas until the end of 2019, beginning of 2020, where it started to prop off on that eastern end and on the western end of St. John, and then has slowly progressed across St. John through the British Virgin Islands, as well as down to St. Croix, which is a good bit south of this map. And then finally, we get to where my thesis lies: stony coral tissue loss disease and thermal stress. So this is a little bit of a debate in the literature currently. So there's been some conflicting data. There's one camp that says there is no relationship between the disease and temperature. One study, Muller et al., looked at the spatial epidemiology of the disease along the Florida Reef Tract, and they found that sea surface temperature wasn't related to disease presence or the density of diseased colonies, which are the two factors that they were really looking at there. And then Williams found that in their looking at the fine scale dynamics of the disease in the lower Florida Keys, they found no relationship between sea surface temperature and the average progression rate. It should be noted, though, that they did find a relationship between the number of degree heating weeks, so the number of weeks above the monthly maximum temperature of the region, and the disease progression, which was a negative relationship, as was found in a couple other studies. So on the other side is those who have found that negative relationship. Myling et al. Out of UVI looked at the dynamics of the disease across a thermal stress event, and they actually found that it slowed or even stopped fully alongside a bleaching event. And then another study, Sharp et al., found that in their looking at the small scale epidemiology of the disease in the Middle Florida Keys, that the disease incidence slowed during the seasonal coral bleaching, but this wasn't repeated in 2019, which was a lesser bleaching year than 2018 for them. And so my thesis and my experiment lie kind of figuring out what the truth of the matter is in here. And so now we're going to move on over into my methods. So we'll start with the experimental design, look at the coral collections, the experimental procedure, and the data analysis. So up first is that experimental design, which includes those colonies of Orbicella faveolata that were chiseled off of the reef, and those big stacks of coolers that were taller than me that I dragged through Kmart on St. Thomas. So the objective of this experiment was really to determine what that relationship between thermal stress and the transmission and progression of the disease was across multiple genotypes of the Caribbean coral Orbicella faveolata. I don't know why that decided backwards there. So how I did that was there were two sets of treatments. There was a diseased set and a non-diseased or apparently healthy set. From there, in each of those were 3 different temperature treatments. There was a control at the normal maximum temperature for the region, an epistress at the bleaching threshold for the region, and then hot, something that should be totally uncomfortable for these corals. They should not be happy. There should, in theory, be some paling and bleaching happening there. And so those temperatures were 28.5, 29.5, and 31 degrees Celsius, respectively. And then within each of those, I had 5 different replicates for a total of 30 coral tanks altogether. So here's the design plan. Up top are some header tanks that were temperature controlled that distributed water down to my experimental units. The experimental tanks sat in a big water table, and I had 3 of those, each with 10 tanks in them. And then the tanks themselves, as you can see over on the lower right-hand side there, had an outer tank that was a water bath for it, and then an inner tank that contained the corals. What that looked like in practice is this. So that bottom left-hand picture there shows the straight-on look at one of the water tables. You can see those bins with the water bath and the smaller inner container there. That terracotta-colored tile in there was used to help weigh down the inner bins because corals were not quite heavy enough to do that themselves, and I did not want them floating around and tipping during the experiment. You can see those tubes that are delivering water, that temperature-controlled water from the coolers, the header tanks, down into the water baths. And then there are also 2 PVC pipes running the length. Those distributed some ambient water drips down into the tank with the corals in it, because corals do not like when their water is kind of stagnant and stale, but it still allowed the diseased water to kind of sit in that tank to allow for transmission, hopefully at that point. And then that photo on the right-hand side, you can see the lovely shelf that Tyler built for me. Thank you. And in that white bucket with the cooler lid on top sat the Apex controller, which I used to basically run the temperature header tanks. They had temperature probes and heaters, and the unit would say, okay, it's at this temperature, I need to turn the heaters on, or it's too low, need to turn, or too high, and I need to turn the heaters off. So that's really what this looked like. Moving on to my coral collections, here you could see 2 baskets of corals waiting to be picked up. So I had 2 different collection sites. The recipient corals, the Orbicella faveolata, were collected from the western side of Brewers Bay on St. Thomas, so right across from where the university and the Marine Science Center are. So it was a nice, easy trip. From there, I collected 15 genotypes, and 4 of them had to be removed from the experiment due to spontaneous disease appearance during that 20-day acclimation period. That does sometimes happen. They get a little stressed as we move them and cut them, and so that can cause their immune system to go a little bit down and allow for diseases that may have otherwise been unnoticeable to infect the corals. And then as for the donor collection, that was done over in Coral Bay on St. John off of Johnson's Reef. That was because the disease was active over there. The disease, by the point I was collecting corals, had become really endemic and chronic on St. Thomas, but there were active lesions over in Coral Bay, and so that was what we used for the experiment. And like I mentioned earlier, these donor corals weren't OFAB, they were actually Orbicella annularis, because that is what we found the most of diseased over there. And then those corals were fragmented into little pieces that were roughly 10 centimeters by 10 centimeters, like this piece here. And they were cut using a chop saw and a tile saw, the former of which you can see Sonora using in the picture on the left-hand side there. And then the letter identifier of the colony was written on the back in Sharpie. In this case, it was J. And then some super glue was put over that so that it did not rub off. I had an awesome team of people that really helped me out with this, thankfully, because it took a long time, even with all of us there. So moving on into my experimental procedure, featuring my experimental notebook that has seen more than its fair share of wear and tear. So I did twice daily observations and photographs of these corals roughly every 12 hours. And what I was looking for was bleaching and paling, signs of disease, a percent mortality, and then any signs of stress. So corals, when they're nice and happy, will kind of put their polyps out, let their tentacles kind of float around. But when they're stressed, they're not going to expend the energy to do that. And then any qualitative notes like the water was super cloudy, or there looks like there's some funky algae growing in the corner. Anything like that that I thought might be important later on. In addition to these twice daily observations and photographs, I also used the PAM Fluorometer to look at the chlorophyll fluorescence. So what this bad boy does is it shoots a light at the coral, and in response, the symbiotic algae and the chlorophyll within it will fluoresce back based on the light. And so what it does is it measures that response fluorescence. In order to do that, the corals need to be dark adapted for an hour. And because of the location of the water tables, it meant it had to be an hour after sunset. And then I did have some procedural changes throughout the experiment. The first and probably most important had to do with the temperature. On the 3rd day of the experiment, I was noticing that the temperatures, particularly in the hot treatment, weren't holding where I wanted them to. And so what I did was take the corals, or take the heaters out of the header tanks for those and actually place them directly in the water baths for each of the hot treatments, and move temperature probes in to make sure that they were working at the right temperature. Unfortunately, due to supplies being limited on consequences of being on a small island, I wasn't able to have enough heaters for the epi-stress treatment, so that got moved down to pretty much the control temperature. But that ended up working out well. The temperature went back up to where I had hoped it would be for the rest of the experiment. Also on that same day, it was a great day for me. The PAM fluorometer had a malfunction during my measurements, and so I wasn't able to take all of the measurements that night. I believe it was a battery issue, but thankfully that was the only day that that happened on. There was also one case of excessively rapid donor fragment tissue loss. And so what I did there is one of the donor fragments in another tank had actually been split in two during the fragmenting process. And so I took one of those two from that tank and put it into the one where the donor fragment had lost all of its tissue. That way, all of the tanks had active disease lesions for the same amount of time. And finally, my controls got disease. So a consequence or potential consequence of taking apparently healthy corals from a diseased, from the same reef as diseased corals is that there could be subclinical levels of disease in those apparently healthy corals, which was the case here. Of the 33 recipient corals in the non-disease treatment, only 6 actually didn't get diseased. So unfortunately, my controls not quite as controlled as I wanted them to be. So just looking at a timeline of this, my experiment started the evening of the 9th, and that was when those diseased donor corals or the apparently healthy donor corals were placed into the treatment tanks. And I counted that day as day 0 because it was the evening when that all happened. The 12th was when I had that heater design change and the PAM failure. The 13th is when I shifted the donor fragments. The 13th is when I shifted the donor fragments. The 15th is when I removed all of the donor corals. That way they had all been exposed for the same amount of time to an active lesion, and none of them had died out sooner. And then the experiment ended on the 19th. Then we went on into the data analysis, where I did some statistics. Who would have guessed? So when I was looking at the transmission, those data were normal, and I was able to perform a couple of ANOVAs to look at the impacts there. For the progression, I actually standardized that data by the first sign of disease. So the day before, the observation before, when they looked like they were totally healthy still, I put as -12 for 12 hours before disease, and the first sign of disease was hour 0. On that data, I ran a non-parametric repeated measures linear model, and then I also took the slope of the line of best fit for each coral fragment and did a Kruskal-Wallis test and then some Mann-Whitney-U post hoc tests. As for the correlation, I did a Spearman's row correlation test because once again, the data for the progression were not normal. And then I also did a non-parametric repeated measures linear model on the fluorescent yield. And I actually adjusted that data as well to the change in fluorescent yield because different coral fragments are going to have different natural yields. So that way they were kind of all starting on the same page. Moving on into my results in discussion, we're going to talk about the spread of the disease, what the temperature treatments looked like, the transmission, progression, correlation, and finally the fluorescent yield. So first up is the spread of the disease. And so what I did here is I wanted to show you what the disease kind of looked like through the time periods. So I have 2 different examples on this slide. The first being a longer term and the second being a shorter term, and those are both very relative. So on the 12th in the evening, that coral looked totally apparently healthy, a little bit stressed, but not doing too bad. The next morning, there were signs of disease, and by that evening, that disease had about doubled in size. The next morning, it had almost doubled in size once again, that size of the mortality area. And then again, that night, it had once again grown. And then by the next morning when I came in, it was completely gone. The little bit of brown you see there is the dead tissue that hasn't completely sloughed off yet and is just kind of sitting on the coral. As for the shorter term one, on the evening of the 11th, again, looked totally apparently normal, a little bit stressed, but overall not diseased. The next morning when I came in, there was definitely two different lesions there. And you can actually see what looks like sand suspended in that is actually the dead tissue. And then by that evening, it was completely dead. So it was a very, very quick progression for that one. So comparing that to another study that looked at Orbicella faveolata, this experiment had corals from the USVI in transmission exclusively through water column suspension. And I found an average of 3.9 days to transmission and an average progression rate of 8.9 square centimeters a day. The experiment from Florida that also looked at Orbicella faveolata took their corals from the Florida Keys and had transmission through direct contact with diseased corals. They had an average of 14.7 to 20.7 days to transmission based on, they looked at different distances from the lesion, and then an average progression rate of 1.2 square centimeters a day. So the disease spread in my experiment seemed to go a lot faster than what they had found over at Mote Marine Lab in Florida. This is what my temperature treatments looked like. For whatever reason, the data from 0 to 24 hours were not logged. I do not know what happened to that data, but you can see by hour 48, it had really not changed too much. They were pretty consistently close. And so that day is when I changed the experiment mode and had that heater design change. And you can see from there, the hot temperature definitely goes up, and the epistress, that yellow, and the control temperatures stay pretty close together and actually were not significantly different from one another. I did include them as separate treatments in my analyses because I didn't know if the different initial starting temperatures would impact or change what that looked like. So moving on to transmission. Across the x-axis there, we have the time of the initial disease observation. So that's how many hours from that start time it took before disease was seen. And then across the y-axis are the number of replicates. So as you can see, there's a pretty decent spread throughout. On average, the hot treatment happened a little bit sooner than the rest, but it really wasn't a significant change in how long it took to get diseased. So kind of looking at that, there wasn't any significant effects of sea surface temperature on the transmission of stony coral tissue loss disease, and there also wasn't any variation among the genotypes. Like I said, the hot treatment tended to transmit faster than the epistress, which was faster than the control on average. But again, that wasn't significant. And more replication of this experiment could potentially isolate and see whether that is really a trend that continues or if that was just kind of how the data worked out in this experiment. Among the treatments, the p-value was 0.909. There we go. So it wasn't significantly different among the treatments or among the genotypes. Moving on over to the progression, as you can see, they're all at 0 at that -12 time frame along the x-axis, which is the time since the initial transmission. And then the y-axis there is that percent tissue loss. Not all of the corals were allowed to go to 100% mortality. A lot of them were actually frozen after they had become diseased in order to preserve any potential microbes and genetic expression for further experimentation. But you can see it's a little bit all over the place. In general, the blue, so that control treatment, was a little bit faster than the epistress treatment, but other than that, it was a lot all over the place, which was actually shown in those results. So there wasn't a significant difference between the control treatment and the hot treatment, or the epistress treatment and the hot treatment. which seemed to suggest that sea surface temperature didn't affect the progression of the disease either. And once again, there was no variation among the genotypes, which might mean that genetics play less of a role in the resistance of a coral to stony coral tissue loss disease, or that there isn't too much genetic diversity at the Brewers Bay Reef site. Either of those could be potential, and looking actually at those results and that DNA would tell us a lot more about that. Interestingly, for this one, the control and epistress treatments were significantly different from one another, even though the temperature treatments weren't. And so once again, that's another place where further replication would really help to identify if it was something off in this experiment or just happened to be that the corals that were in there reacted a little bit weirdly. Looking at the correlation between transmission and progression. So across the x-axis there, we have the time to the initial disease progression, just like we saw in the transmission graph. And along the y-axis is the slope of best fit of the line of best fit for the disease progression. So it follows a little bit of a pattern, but it's spread enough all over the place that the results of the correlation were insignificant. So pretty much there wasn't really a correlation between the time to the initial disease transmission and the progression slope of the line of best fit. So pretty much how long it took the coral to become diseased didn't impact how quickly the disease worked its way through the coral. Finally, we get to the fluorescent yield. Across that x-axis is the time since transmission. I have a black vertical line at the 0 hour because there's a bit of a skew, so you can have an idea of where that disease transmission was. And across the y-axis, we have that fluorescent yield. As you can see, it's kind of all over the place. There's not really a difference across time or among the treatments. And so I really found that there wasn't a difference in the change of chlorophyll yield across time among treatments or among treatments across time. And this could be because of either the speed of the progression, how I PAM'd. So what I did is I actually moved the sampling across the healthy side of the lesion and kind of followed the lesion back. So all of those measurements were from the apparently healthy side. And so because of how rapidly the disease moves, it may not have started to really impact there yet. And also the endemic disease state of Brewers Bay, they might be a little bit more resistant and reluctant to release their symbionts in the face of this disease. So wrapping this all up, we're going to go through in summary and where we go from here. So sea surface temperature is likely not the factor that drives the negative relationship between thermal stress events and the transmission and progression of the disease. So like I said earlier, the Williams et al. Study from 2021 found that sea surface temperature wasn't didn't have a significant relationship, but actually the degree heating leaks did. And those studies that found that negative relationship looked more at a thermal stress event rather than specifically temperature. And so that leads me to believe that there is probably another factor that is correlated with a thermal stress event that is what's causing that disease to slow down a little bit. And so further studies into kind of what other factors coincide with thermal stress events will give us that answer of what is actually causing the disease to slow down, if it does. And so what now? Like I said, there are a lot of future studies that can be done on this disease, from the thermal stress event research that I was mentioning, to even just the pathogen of the disease and possible treatments. and that sort of thing. This disease is spreading through the Caribbean, but there is a worry that given the global world we're living in, it could spread beyond the Caribbean. And so knowing as much as we can about the disease is going to be really paramount to our ability to respond to both it and future coral diseases. It can also be used for reef management. So if we figure out what is causing the disease to slow down during thermal stress events, we could identify those time periods where the disease is going to slow down and potentially use those to catch up with treatments to the diseased areas. So using the antibiotics and the probiotics that have been found to kind of help stop the diseased lesions, we could use that little bit of a respite to try and catch up to where the disease has already been spread. Also, coral conservation support. Thermal stress is not going away, and neither are coral diseases. This one is actually probably the most virulent coral disease on record, and it's probably not going to get better. Coral diseases will continue to happen. Thermal stress events are going to continue to happen the way our world is going right now. And so support for corals conservation is going to become increasingly important, especially to that 500 million people and the 25% of ocean biodiversity. And then finally, we as individuals can take the small steps because it's not going to happen in a great storm all at once. It's going to be the little things that bring us to a healthier ocean. So before I wrap this up and go to questions, I have some people to acknowledge. First and foremost, my sincerest gratitude to Dr. Tyler Smith and the rest of my thesis committee, Marilyn Brandt and Carlos Prada, for all of their help and guidance throughout this experiment and project. Big shout out to Aurelie Chaparro, who helped me with my setup. It was a long time of just the two of us working at that water table. She also did the disease experiment before me, so we bounced a lot of ideas off of her. And so it was really helpful to have her around. Also, a big shout out to my coral collectors and my coral fraggers. I couldn't have done that in as nearly as a timely a fashion without you guys. And then, of course, to the funding sources, the Lonavento Charitable Trust through UVI and VI EPSCoR as well. But yeah, that brings me to the end of this presentation. Across the top there, you see some photos of the non-coral side of my experiment, from Tyler up on top of the water tables, fixing something for me, to my cat, who was very upset that I was gone at least twice a day, my candle of a prayer for useful data, which jury's still out on whether or not it worked. One of the many, many beautiful sunsets I watched waiting to PAM at the university. And then finally, my spare tire that waited 2 days after my experiment ended to explode. So I only had to change my tire once during my experiment. But that about wraps it up for me. And I am going to open this up for questions.
Very good, Reagan. Yes, if anyone has any questions, just feel free to use your reactions to raise your hand, and I'll go to a different view. You can see people. Any questions, anyone? If not, Reagan, I have one to get started. You mentioned the observational evidence that you had, and I'm wondering what, if anything, you saw in that evidence in terms of bleaching, in terms of some of the other metrics that you were looking at. Was anything useful, or any difference between the heat and not heated treatments?
Yeah, so there was definitely a little bit of discoloration that I saw throughout. But for the most part, the disease progressed rapidly enough that I wasn't really able to see much difference. And all of the corals appeared equally stressed throughout the time period. It was June, and so it was definitely warmer, but no, I didn't really see any differences there.
Okay, thanks. Teresa Turner, you have a question?
Yes, I was wondering, do you have any idea what species of zooxanthellae were in the corals?
I do not. We didn't end up getting to look at that. Given where they were collected from, I believe it's likely that they were clades A or C because they came from the kind of parallel to the ocean surface and not that side where it might be a little bit less bright and where the clade D tends to like to be.
Do you still have the samples so somebody else could look at them?
Yes, I do believe that those samples are still down in the freezer. I believe Marilyn would know more about what's happening with those.
And the other question I had, again, I think you didn't do this, but if there's any plans to get like density of zooxanthellae instead of just the PAM to look at the bleaching part of it?
Yeah, no, unfortunately, that wasn't something that I looked at during this experiment, but that's definitely something that future experiments could look at and see if that side of it really helped or was impacted at all.
Thank you.
Nicole, you have a question?
Yeah, I do. Hi, Reagan. First of all, great presentation. I have a question about the fact that you had different donor and recipient sites. So being here, obviously, I understand that there were probably no active lesions for your donor sites. That means like you're giving the disease, right? They're the disease colony.
Yeah.
Is there a reason why you still chose to collect corals near St. Thomas when you know that they have had the disease for like a year and a half? So likely the corals that you're collecting are I would say more resilient to it since they've been able to survive thus far. And did you see that in your data, or can you speak a little bit to that?
Yeah, of course. A lot of that came with kind of the ease of transportation and the availability of people for collectors. So those recipient corals that were collected from Brewers Bay, it was just Tyler and I collecting. And we had a bigger team for the St. John collection. And because I was looking specifically at Orbicella faveolata, which tended to be, I believe, a bit more in the Brewers Bay area than the Coral Bay area. And also I didn't necessarily want to take corals from the same, the recipient corals from that same reef in hopes to avoid what happened with my non-disease controls, where they actually had subclinical infections when they were collected.
Right, yeah, that makes sense. Thank you.
Yep.
Any other questions? Oh, Larry?
Hey, Reagan, great talk. I'm curious if perhaps Orbicella might be a little more temperature resilient than some of the other coral species on the reef, and maybe the anecdotal evidence we have for this temperature dependence might be the case in some other species out there. I wonder what you thought about that.
Yeah, that's definitely a possibility. Orbicella was chosen partially because it was kind of in that mid-range of disease susceptibility, so it wasn't super susceptible, will definitely get diseased super easily. It was a little bit more resilient, and I didn't find necessarily anything that said that they were more resilient to bleaching and thermal stress events. Nothing in the literature seemed to show that. They seemed to bleach somewhat regularly alongside other corals that were the similar mounding corals.
Good to know.
Well, that's great. I mean, I have more questions, but I will save them for the closed defense. If there are no further questions, I think we can thank Reagan for a lovely thesis. And then in about a couple minutes, we can retire with the committee privately to do the rest of the meeting.
All right. Well, thank you all so much for being here. It was so great to have you all here for my thesis defense. And quickly, I click on the right thing. Here are my references. There are a lot of them. And the sources for the photos that weren't taken by me and the figures that weren't created by me. But thank you all so much. And I hope you had a great time.